This is a podcast of the Nuffield Department of Medicine. Today we speak with Professor Panagis Filippakopoulos about his research on targeting epigenetics to treat cancer. Hi Panagis.
Good morning.
Can you tell us about DNA transcription?
DNA transcription is a very complicated process that involves the segregation of factors that come to basically duplicate the genetic information in order to generate the tools necessary for the cell survival. And this process is initiated by chemical modifications, chemical signals that are somehow interpreted by evolutionary conserved modules that basically segregate the machinery at a specific locus where transcription is going to be carried out. And this process is often deregulated in cancer.
And what's the focus of your research group?
We are interested actually in understanding how the assembly of the transcriptional machinery is initiated by this readout process, this recognition of these chemical modifications by these evolutionary conserved effector modules. And among other things, what we do is we generate small molecule inhibitors that are used to block this interaction, this recognition process, in order to study what happens when the process is disrupted, like in cancer or in other diseases. Using these tools, basically we can understand how a normal cell functions and understand also how this is perturbed in disease.
Can you give us an example?
We are interested in a rare but extremely aggressive tumor type termed non-covalent carcinoma, which involves a genetic fusion between a protein that normally expresses in the testis, and somehow this fusion results in expression of the protein in the mediastinum. It's very aggressive, it's deadly. We do not understand the molecular mechanism, but what we do know is that it hijacks this process of readout, basically taking advantage of it and sequestering the transcriptional machinery to propagate and enhance tumor growth. But the important thing is that it can be targeted by small molecule inhibitors. The problem with non-covalent carcinoma is that there is no easy detection. So in principle there have been no cases in the UK, and it's only been detected in the US. So what we're interested in is understanding the molecular basis of the malignancy in order to define the biomarkers to be able to detect it and eventually cure it.
What are the most important lines of research that have developed in the past 5 to 10 years?
Well, there are many significant things that have happened actually, but I think the most exciting for us is the fact that the recognition process of the chemical interpretation, if you wish, of signals can be directly inhibited by small molecule inhibitors. So this has already been translated to the clinic, at least in an attempt to cure this rare tumor type, the non-covalent carcinoma, but also it paved the road to look into the deregulation of this signal readout process in general and define mechanisms in other diseases. And I mean, probably the most important aspect of that is that in the last maybe 5 years, there's 12 clinical trials that have been initiated using such chemical tools.
Why does your line of research matter? Why should we fund it?
What we do is considered basic research, but in principle it allows to understand and define the molecular mechanisms underpinning disease. And by doing so, we define the biomarkers necessary for detection, but also we help understand how can we make safer drugs more effective, more selective, avoiding cross-reactivities and off-target effects. And often it is translational to the clinic.
So this is how you feel it fits into translational medicine within the department?
Absolutely. I think that medicine goes hand in hand with basic research. One helps the other. We are embedded in a department where we have access to clinical material, samples, expertise, ideas and opinions. So in principle, we help define the molecular mechanisms underpinning disease and also find biomarkers that can help better understand how to target disease in the clinic. And also generating key tools that can be used eventually as lead compounds to translate directly into the clinic.
Great. Thanks, Panagis.
Thank you.